Hi, everybody. I'm Mark Musin from Stanford University, and I have the pleasure of introducing our speaker, Alex Diehl. Alex is a biologist and an ontologist, and he's an associate professor at the University of Buffalo. Biologically speaking, he got his PhD in molecular and cell biology from WashU, did stints at Karolinska, was part of Decode Genetics, and then went to Jackson Labs for the mouse genome informatics group, where he got really hooked on ontologies. And now in his career at Buffalo, he's been involved in a large number of ontologies that are important for biomedicine. He's particularly involved in the cell ontology, which we're going to hear about this afternoon. Alex is going to talk about the cell ontology and its role in organizing high-dimensional cell type data. Yes, Alex, we're looking forward to your talk.
Thank you, Mark. And here we go. Can you see my screen?
Not yet.
Oh dear.
Now we see your screen.
You do? Okay, thank you. Okay, great. Thank you, Mark, and thank you, Kadi and the HuBMAP Consortium for inviting me here today. I'm very pleased to speak to you about the cell ontology, and I'm very pleased that you have already incorporated the cell ontology in your workflow. I'm going to give you some background on the cell ontology. I know many of you are aware of it. I think it's useful to know a bit of the detail behind it. So the cell ontology obviously provides an ontological representation for cell types across biology. And it's around 2,200 cell type classes at this point. It serves as the designated high-level ontology for cell types in the OBO Foundry. And I'll talk about the OBO Foundry a bit in a moment and why I think that's important. But the majority of the content of the cell ontology is actually focused on vertebrate cell types with a strong emphasis on mammals and humans. And by agreement, other communities of biologists work on different cell types in a compatible manner within with the cell ontology within the context of the OBO Foundry. So for instance, the plant ontology people handle the plant cells, the Drosophila gross anatomy people handle Drosophila and insect cells, and there's also, for instance, a separate ontology for cell lines, which is linked into the cell ontology. I'm just providing here some relevant URLs. You can always download any OBO ontology with a URL of this format shown at the top here. And the OBO Foundry maintains an information page. There are multiple browsers here. OntoB, BioPortal, and the Ontology Lookup Service are all three good browsers for browsing the cell ontology. There's a GitHub repository we maintain for our work on the cell ontology. And we, of course, primarily use the Protege editor when we edit the ontology. So cell ontology is a project that really grew out of the gene ontology. Gene ontology, of course, is now over 20 years old. Cell ontology is 15, 16 years old. I became involved in 2005, really the first major revision of the cell ontology for immune cell types, which I carried out. The cell ontology is one of the founding members of the OBO Foundry. And throughout the development of the cell ontology over the years, we've introduced a logical rigor, typically in the form of logical definitions that provide background to cell ontology terms, and I'll discuss that in a moment. And since around 2012, the cell ontology has been playing a part in data analysis by major scientific initiatives such as Phantom 5 or ENCODE. or ImmPort. Right now, we're starting a major revision of the cell ontology to improve the representation of species-specific cell types. So the OBO Foundry is a collective of ontology developers who are committed to collaboration and adherence to a set of 16 shared principles, including open use, collaborative development, non-overlapping and strictly scoped content, and common syntax and relations. These principles ensure that these ontologies are interoperable and for the most part, non-redundant with each other. And most OBO Foundry ontologies are compatible with the upper level basic formal ontology and employ relations defined by the relations ontology. And this is important because I note that HuBMAP also uses Uberon, which is another OBO Foundry ontology. And Uberon and the cell ontology are developed really hand in hand with each other. Cell ontology terms typically reference Uberon terms when referring to the anatomical location of a cell type, for instance. The OBO Foundry actually maintains its own website, and there's over 100 ontologies listed here. Some of these ontologies have actually gone through a review process to ensure they're really adhering to these 16 principles. But overall, all of the people who are listed, all the ontologies that are listed here, rather, are committed to working together, and it really makes it for a very good community. I'm going to briefly talk here about what an ontology is. So an ontology is a formal representation of the entities that exist in reality, that are of interest to a field of knowledge, and the logical relationships that exist among these entities, as well as between these entities and entities in related domains. And this is important because ontologies are not the same as terminologies, nomenclatures, although they may incorporate those things. They're much more logically grounded, and we like to say that they reflect what's actually in reality, not concepts of what is in reality. And this is important because, you know, when we talk about data, we want to be able to interpret that data correctly, and we want to be able to represent it in ontologies in a way that really reflects the biological reality behind the data. So the meaning of ontology classes comes not from their names, but rather the definitions. But it's important that, I mean, careful naming is important because it's an aid to both human users, but also for searching and applications like natural language processing. And we do recognize this. Naming is going to be an issue as cell types become more complex in their definition, because the simple names are basically well known and taken, but we need to be able to create names for subclasses that might reflect molecular markers or other aspects of the cells. So ideally, ontology classes will have both textual and logical definitions, and the axioms of these logical definitions allow for reasoning across the ontology to infer implicit relationships among the entities. So as I mentioned, ontology is not a terminology or nomenclature, although it may reflect the accepted terminology or nomenclature of the field. It's not a random collection of anything with a connection to a field of knowledge. In other words, there's an organization to it, and there's a scope. And it's not a database or knowledge base, meaning that ontologies are not intended to collect all information about a particular area. The cell ontology is not intended to be a database about cell types. Ontology classes are typically defined with the necessary and sufficient criteria and do not capture all the facts or knowledge that is known about an entity. So domain ontologies focus on a single area of knowledge, and in anatomy, the representation of entities is split among multiple domain ontologies according to levels of granularity and type of entity. So whether something is a material entity such as an organ, or for instance, something is really a biological process that occurs in the entity. We have the protein ontology covering proteins, including isoforms and modified forms, and specific representations of protein complexes. The Go cellular component provides a representation of parts of the cell. This includes high-level representations of the protein complexes. Now, in fact, protein complexes are represented in the protein ontology typically with reference to those that are defined in the Go, but in the protein ontology, they're really defined according to their component polypeptide chains. The cell ontology obviously covers cell types, uberon, tissues and organs, anatomical structures, and the gene ontology biological process ontology covers biological processes occurring at any level of granularity. So application ontologies bring relevant classes together from multiple domain ontology to provide an ontological representation of a complex area of knowledge. So in HuBMAP, you have the CCF semantic ontology. It's really an application ontology that includes relevant classes from uberon and the cell ontology, as well as classes for biomarkers that are needed to describe anatomical parts and cell types, which are under study by HuBMAP. As people develop these application ontologies, generally the best methodology is to request new classes from core ontologies for relevant entities not currently represented in those ontologies. So how do we represent cells in the cell ontology? Well, typically we reference things like morphology, surface marker expression, singly or in combination, and in some cases we actually reference lack of a cell surface marker, which is important for definition of particularly immune cell types. Transcription factor expression or expression of other internal proteins or genes, singly or in combination, by lineage and by function or capability. Excuse me. These definitions, the definitions of cells come from multiple lines of evidence, including microscopy with or without staining, immunofluorescence, flow cytometry or mass cytometry, colony formation assays, in vivo or in vitro lineage tracking, direct assays of cellular function, indirect assays of cellular function, assays of gene expression, such as single cell RNA sequencing. So I'm just going to go through a few examples here. So when we consider the cell type induced T regulatory cell, we have a definition that's very explicit here. It's a subclass of CD4 positive alpha beta T cell with the phenotype CD25 positive, CTLA4 positive, and FOXP3 positive with regulatory function. And we provide typically a number of synonyms. Some of these synonyms are simply lexical variants, like whether or not we have a hyphen. In other cases, we try to capture names that are used in the literature to refer to the same cell type. In some cases, these are what we call related synonyms, meaning that there's a term in the literature that may refer to this or perhaps some other cell type at the same time. And we always provide a definition, a source rather, for the definition. So typically a PubMed ID for a paper. We also, not noted here, but we always include the ontology editor. Used to be this was primarily with the initials of the ontology editor, but now we've moved to using ORCID IDs, which is really a much better way of really identifying people. So we can look at the representation of induced T regulatory cell in this sort of graphical format. We see it's a subclass of CD4 positive, CD25 positive alpha beta regulatory T cell, which itself is a subclass of CD4 positive alpha beta regulatory T cell. Now it develops from an activated form of CD4 positive alpha beta T cell. Because it's a subclass of the CD4 positive alpha, CD25 positive alpha beta regulatory T cell, it actually inherits the has-part relationship here pointing to the interleukin-2 receptor subunit alpha, which is otherwise known as CD25. This just points out the fact that sometimes naming is complex. This name is the name used by UniProt and the protein ontology for CD25. We know that the name we choose here is important for immunologists primarily, and the CD marker names are more commonly used. And so we have to point to this class that's named differently, but we understand that it's an appropriate class because it is the same as CD25. Now, of course, there's additional criteria here that the induced T regulatory cell has CTLA4 and FOXP3. And again, these names are written differently than we would write in shorthand FOXP3, for instance. But again, that reflects the fact that ontology names are sometimes formalized in different communities according to different criteria. So we capture this in the form of what we call an equivalence axiom or a logical definition. So we say that induced T regulatory cell is equivalent to a CD4 positive, CD25 positive alpha beta regulatory T cell. It has part FOXP3 again. It has a plasma membrane part CTLA4, and this relation designates that the CTLA4 is actually at the plasma membrane. And it develops from the activated CD4 positive alpha beta T cell. It inherits these axioms from its superclasses, basically. So it's part of some immune system. It develops from hematopoietic stem cell. And it has the T cell receptor, for instance, and so on. So the cell ontology, or CL as we call it for short, uses relations to connect to external ontologies in order to form these logical definitions. And these include capable of, when we want to refer to a gene ontology, molecular function, or a gene ontology biological process. Quality or relation when referring to a biological quality that might refer to, for instance, some aspect of the cell morphology. And we use the protein ontology for basically representing proteins that may be part of a cell, either on the cell surface or internally. We use Uberon to specify what the cell is part of anatomically. We sometimes use located in if, for instance, immune cell types, which perhaps are only transitorily in a particular tissue. And we use the Go cellular component ontology for other types of internal parts that may be important for defining a particular cell type. So for certain hierarchies of cell types, we have quite good representations, or fairly in-depth representations. So for instance, kidney cell, we have 126 subclasses. This includes things like kidney epithelial cells, of which there's 101 subclasses. And hopefully this serves the needs. There's been discussion, particularly with folks in KPMP, about whether we're actually over-specifying here. But I think we've made the decision to keep this representation while we hear back from the work of KPMP and hopefully HuBMAP too about what cell types are truly important here to represent. So for instance, we have this hierarchy of epithelial cells, which gets quite detailed. However, the logical definitions here are much simpler than for the induced T regulatory cell. In that, in this case, we're really only talking about an epithelial cell that's part of some kidney. And so it doesn't include any information about markers that might be important. It doesn't include any information about any other aspect of the cell that might be considered important. It seems like the most relevant aspects here are simply the fact that it's in the kidney. A more granular cell type here is kidney cortex collecting duct intercalated cell. And again, it has a fairly simple logical definition. It's a renal intercalated cell that's part of some cortical collecting duct. We can reflect this graphically by this external relationship to the Uberon term cortical collecting duct, which is again part of the cortex of the kidney, which is part of the kidney. At the same time, we have a sort of multiple inheritance pathway here about actually what cell type, what parents of this cell type are, and that's because depending on different criteria, we can sort of classify cells in sort of different ways. So we have kidney cortex collecting duct epithelial cell versus kidney collecting duct epithelial cell. And this one is probably more specific than this one, but it's really having this level of detail may help when we're sort of pinning down like RNA, single cell RNA-seq results to a very specific location in the kidney. So, one of the great things about using ontologies in this way is that we can really make assertions about, for instance, where proteins are expressed based on the fact that we can go from the molecular level all the way up to the whole body, to all the way up the anatomical tree here. And so these folks published a paper in 1997, which was then converted to a gene ontology annotation. The ontology annotation, gene ontology annotation states that the aquaporin-1 protein is located in the brush border membrane. And so we can capture the fact that this protein with a protein ontology ID is part of the brush border membrane, which is specified by the Go cellular component term, which itself is part of the brush border, which itself is part of the brush border cell, which itself is part of the proximal tubule, part of the nephron, part of the kidney, urinary system, all the way up this level of granularity from a very small size to sort of the whole body. And so we can query, use this knowledge to query and say, if we were interested in all proteins expressed in brush border cells, aquaporin-1 would theoretically be returned based on this type of relationship. So there's some considerations here for use of the cell ontology in data analysis. Cell ontology has good general coverage of cell types in human, mouse, mammals, and immune cell types are particularly well represented with extensive logical definitions tied to cell marker expression and other characteristics. There are 593 classes of hematopoietic cells, which include 461 classes of leukocytes, for instance. And this is going to grow because of the continuing interest of the Human Immunology Project Consortium, as well as my own personal interest in immunology. Cell types of other major organ systems are fairly well represented, though not exhaustively. So for instance, we have 333 nervous system cell types, 126 kidney cell types, 123 digestive tract cell types, 70 skin cell types. But for instance, there's only 10 liver cell types and 3 gallbladder cell types. And I suspect there'll be more needed to represent the data coming from HuBMAP and other sources in the future. We recognize that we still need robust logical definitions to reflect current knowledge for many cell types. And as I say, we're working on improving species-specific cell type representation. It's important, particularly among immune cells, because similar immune cells in human and mouse, for instance, are often defined using different markers, even though they share characteristics like their functional activity. Plasmacytoid dendritic cell is one example of this. So these immune cell type markers are present for many of the cell types in the immune cell hierarchy. And we started adding these 10, 11 years ago. And it was recognized that that this could be the basis of using the cell ontology as an adjunct to flow cytometry analysis. So Ryan Brinkman and his group developed a tool called FlowCL, which takes the results of automated gating of cell populations and assigns markers, and then queries the cell ontology triple store to match identified populations with their combinations of markers to known cell types, and then returns the cell ontology class. So I mean, this is obviously very useful, and it's been built into the ImmPort Galaxy system, which is a system that the ImmPort consortium has built based on the Galaxy framework. The ImmPort Galaxy set of tools are designed to facilitate flow cytometry analysis, and part of that is a workflow where you can load FCS data into the ImmPort Galaxy system, use the flock algorithm to cluster the data, and then the output of that is then transferred to this FlowCL tool, which queries the cell ontology triple store. And then these cell type results are output, so that when we use flock on a fairly complex flow cytometry experiment, you can typically identify multiple populations, and then those populations can be matched to classes in the cell ontology. So some years ago, my group, we had the idea about whether we could use the cell ontology as a framework for comparing gene expression data that's tied to specific cell types. We want to be able to identify genes that distinguish one cell type from closely related cell type. Now, at the time, this was really pre single cell RNA-seq, so we were using bulk RNA data from the Immunological Genome Project, which provides gene expression data based on sorted mouse immune cell types that are all done according to standardized methods. So we mapped their cell types to cell type terms or classes in the cell ontology. And then we used the cell ontology structure to guide comparisons between cell types. So we compared their expression patterns, gene expression patterns from 88 cell types in a pairwise fashion, resulting in a total of 7,656 comparisons. And we used a sort of ontological framework to map these gene sets to cell ontology classes. So basically, we looked for cases where, for instance, genes were upregulated in a germinal centered B cell, but downregulated in the sibling class here in the ontology, the marginal zone B cell. We did this across the ontology so that we'd had, as I say, over 7,000 sets of comparisons. And these comparisons enabled us to identify cells, or rather genes, that would distinguish one cell type from a closely related cell type or from a more distantly related cell type. This allowed us to determine sets of genes that were upregulated or downregulated in particular broad classes of cells, and also in the more granular classes. And so, for instance, we identified a few novel genes for specific cell types. For instance, these SCD1, an enzyme involved in biosynthesis of monounsaturated fatty acids, whose expression is restricted to mature B cell types in comparison to other immune cell types. And this I83 Recon gene, which is basically uncharacterized, but it's restricted in lymphocytes to marginal zone B cells. So obviously a lot of this type of work can be done with more modern tools using single cell RNA-seq data, but this work, I think, shows how we can use the ontology to structure our analyses. We identified a set of candidate genes that seem to be involved in unique functions of germinal center B cells. And again, some of this has been confirmed by later work. I think it points to how the ontology provides an assumed relationship between cell types, and we can use that then to sort of guide our judgment about how closely these cell types are related to each other, or how distantly they're related to each other. So now I'm going to switch the uses of the cell ontology as metadata for large data sets. I've been involved in the ImmPort project at various levels over the years. ImmPort decided early on they were going to use ontologies as part of their metadata for any data that's submitted. And they use the cell ontology as metadata for any cell type related data. And this allows them to organize data according to the origin of that data, of the samples here. So we have, for instance, T cell and B cell, dendritic cell. But it's also important because when people submit, for instance, flow cytometry data to ImmPort, they need to reference cell ontology classes as part of how they submit that data. So for instance, in this case, they may have a population definition for a particular cell population. And when they submit the data, they're supposed to map that to a cell ontology class. And then if they have additional markers they're looking at, they use a very special defined formalism here, which is simply using an ampersand plus as the marker as part of the submission. The idea here is that we're trying to regularize how data is submitted to ImmPort, so it's easier then for computational manipulation later on. The Phantom 5 project, we contributed the cell ontology as part of the sample ontology that was used to identify the source of RNA that Phantom 5 studied in terms of cell types, tissue, cell lines, and disease types. Cell ontology was a key component of the Phantom 5 sample ontology, and the sample ontology identified the source of all samples. So this included 573 human cell types, 128 mouse cell types. Tissues were handled by Uberon, and cell lines, I think by the cell line ontology. For instance, this was useful because it was clear when they clustered various promoters seen by determining the 5' ends of the mRNAs using their methods. They could see that, for instance, lymphoid cells and mast cells, for instance, clustered close together. All the nervous system cell types clustered together, and epithelial cells clustered close together. So this was very nice because it showed how there's really common patterns of promoters used in these different cell types. Another resource, ArrayExpress, uses the cell ontology as a component of their experimental factor ontology to drive searching. And we can see the hierarchy of the cell ontology here. If you type in B cell, you immediately get this hierarchy underneath here. And so they've standardized all their all the metadata for their submitted gene expression data sets to ontology terms, including the cell ontology. And it really aids in the use of in querying, I would say, array express, because you know, it doesn't matter if people might have put in the original metadata, for instance, B lymphocyte or some other way of referring to a B cell. They're all standardized to the common ontology terms. So we know a deluge of cell type data is on the way, including high dimensional flow cytometry, mass cytometry, single cell RNA sequencing, and site-seq, and enhanced in situ methods for studying gene and protein expression. Obviously, major projects such as the Human Cell Atlas, KPMP, and HuBMAP are generating a huge amount of data here. Mass cytometry analysis often offers a chance to view collections of related cell types that may differ in their marker expression by perhaps a single marker or just a few markers. If we look at this spade analysis here, we see, for instance, multiple variations on CD4 T cells, CD8 T cells, and these can often be tied to known cell types identified by earlier methods. Single cell RNA sequencing allows for identification of many clusters of related cells. And for instance, this experiment on neurons in the mouse hypothalamus, they could identify 50 major cell types via clustering. Now, when we think about this in terms of how we want to go to the cell ontology, we have to consider how many of these are new cell types versus known cell types. Meaning, we can often see the expression of perhaps hundreds of different transcripts in each individual cell, but how much variation is there? Are there really closely related points here which really represent the same cell type? Are there different cell states of the same cell type reflected in these clustering? And if we're going to define novel cell types here, how are we going to name or define them? And I'll get back to that in a moment. Brian Averman and Richard Sherman's group and others have developed this random forest technique for creating necessary and sufficient sets of genes to uniquely define cell types in the brain. So they took basically a nuclei sorted from specific regions of the brain and sequenced RNA, clustered, and then used a random forest technique to really come up with the combination, the minimal combination of genes that could uniquely identify every cell type in that sample. And they could group these neurons that they were looking at based on some known markers into excitatory, inhibitory, and glial cells. And they identified particular genes of interest for each of these subclasses here, for instance, of the inhibitory cells. And they're able to then define sets of 3 or 4 genes that within this particular sample can uniquely identify the cells. Now, a challenge here is that if we take this across the entire brain, we may need more than 3 or 4 genes being expressed to uniquely identify a cell type in one portion of the brain with from a cell type that's very similar, but in a different portion of the brain. There's been tools published recently, and I've seen some more this morning that use the cell ontology as the basis of classification of single cell RNA-seq data. For instance, this tool OnClass, developed by a group, I think at Stanford, uses the cell ontology class to infer cell type relationships in order to classify cells. It actually has a way of possibly being able to refine the cell ontology to add marker genes for certain cell types in the cell ontology where those genes haven't been expressed before. So, I mean, basically, there's a they take the data of the cell ontology and map it onto a low dimensional space here, and then they add in their experimental data, and they can determine if particular cell clusters from their or single cells rather from their experimental data map close to the known cell types from the cell ontology in their experimental space, and this hopefully allows identification of cell types. A similar approach, perhaps, is this recently published cell blast method, which uses a neural network-based generative model to estimate cell similarity in some sort of precise manner. And I think the interesting part here is they developed and rely upon a well-curated multi-species single-cell transcriptomics database called the ACA, the Animal Cell Atlas, which is based on manual assignment of cell ontology terms to single-cell RNA-seq results. And by comparison of experimental data to this manually curated resource, they're able to automatically annotate known cell types and identify novel cell types. So I think obviously people today have presented some similar approaches here, and I assume that these things will be refined as time goes on. So I'm going to finish up here with some important discussion here. So when we consider novel cell types in the cell ontology, ideally cell types identified via mass cytometry or single-cell RNA-seq can be identified as subtypes or subclasses of known cell types in the cell ontology. If we're going to add new cell types to the cell ontology, ideally they should be substantiated by using multiple lines of evidence. So not, for instance, data from a single experiment, but multiple experiments, hopefully coupled with maybe in situ data or other types of data beyond the single-cell RNA-seq. The cell ontology, I will note, serves multiple communities, and all of whom may contribute new cell types or request revisions or improvements to particular cell type classes or the hierarchy itself. And because of this, we have to be aware that we have to do our best to satisfy everybody as best we can. Another interesting thing is that this recent technique of site-seq, which couples cell surface protein expression with single-cell RNA-seq, shows that cell populations defined by protein expression may correspond to multiple clusters based on single-cell RNA-seq data due to stochastic variations in gene expression. This means we can't, or we should be careful not to create excessively granular cell types in the cell ontology based solely on single-cell RNA-seq data, because again, coupling it to knowledge of protein expression, which is more persistent, may just simply show that multiple clusters from the single-cell RNA-seq analysis really identify the same cell type. We know, of course, there are challenges in cell naming and definition. So we need to establish standards for naming of novel cell types. And I think Amy Bernard's going to talk about that tomorrow in the context of cells of the nervous system. So we need names to be meaningful and hopefully agreeable across interested communities. But really good definitions are more important than good names. So we need good methods to elucidate minimal, necessary, and sufficient condition to molecularly define cell types, which ensure that these definitions are unambiguous with regard to similar cell types across the body. So if we want to describe a pyramidal cell in one part of the brain in a very molecular specific fashion, we don't know if it's going to have similar patterns of expression to a pyramidal cell somewhere else. But if we can't come up with different criteria, then that set of genes simply won't be sufficient to distinguish these two types of pyramidal cell. Now, I would note that when we define cells, we often make reference, of course, to anatomical location, and that can be part of criteria when we're considering cell types in a fixed organ, as opposed to, say, lymphocytes. So we also need to resolve conflicts when separate groups submit differing ways of defining what appear to be the same as cell type. This might mean that we have to make a combined definition or create new siblings or subclasses if we can really distinguish two very similar cell types. In terms of the use of the cell ontology data annotation, cell type classes in the cell ontology used to annotate particular cell type data should be determined based both on the name and the definition. Again, definition is a key feature of ontology, and names can sometimes be deceptive or even misused in certain cases. The data about an observed cell or cell population should not contradict the definition of the cell ontology class used to annotate that data. And if it does, then that might be an indication we need a different class, maybe a new class, to be added to the cell ontology. The most granular class in the cell ontology that can be used should generally be picked. But at the same time, we have to recognize that not all criteria of the cell ontology class need to be fulfilled if information is lacking, while in some cases a more general cell type may be more appropriate than a more specific cell type. So as we know, HuBMAP uses cell ontology classes as part of the ASCT plus B tables that enumerate cell types under study. And of course, from the perspective of the cell ontology, we would really like if HuBMAP works with us and requests missing cell type classes from the cell ontology. When HuBMAP protein and gene expression data tied to particular cell types becomes available, in some cases we'll be using that data to enrich definitions in the cell ontology. Now again, the cell ontology is not meant to be a knowledge base for the expression storing the expression of, for instance, all genes that are seen in a particular gene expressed in a particular cell type. But we want to improve definitions so that we can have multiple subclasses of epithelial cells that are distinguished by their patterns of gene expression.
Alex, we have about 1 minute left and plenty of questions for you.
Okay, I'm nearly done. Data query and analysis systems of HuBMAP should ideally take advantage of the linked ontology hierarchies and logical definitions of Uberon in the cell ontology to allow for more complex searching. I just want to mention we have 2 cell ontology events going on later this week, just by chance. We're having a cell ontology training session for users and outside editors. I know there's a few people from HuBMAP who've signed up for this. There's still times to sign up. We're trying to train people to make requests to the cell ontology. This session will help them do that. On Friday, we have the Cells 2020 workshop, which is associated with the International Conference for Biomedical Ontology. We have 4 talks about aspects of ontologies related to describing cell types in different contexts. And I've had the pleasure of working with many talented people over the years, and I thank you all for your attention.
Thank you so very much, Alex, for this fantastic talk. The Q&A section has been buzzing with activity, and I'm going to begin with the first question that was asked by Richard Conroy. I'm also going to post that into the chat here. Alex, is there a way of easily adding spatial that is adjacent to or temporal that is differentiates into relationships to the cell ontology? And Alex, you can also read that question in the chat window.
Is that the first question I see here? Or?
Yes.
We haven't typically collected that type of information for the cell ontology. That's a component, for instance, of the foundational model of anatomy, where they have information not really at the cell level, but at the tissue level and organ level about adjacency and spatial relationships. I think we could add that data for cell types if in particular it's important for definition of certain cell types. So we might consider, cell types in sort of transitional zones where you might say a particular cell type is adjacent to a slightly different cell type in that transitional zone, and that may have consequences for its particular gene expression. So that, again, we can add that information if people provide us with good examples in a use case. Are you going to be reading the questions, or shall I?
Yes, I will. Thanks so much, Alex. I'm going to move on to the next question. I will also post it into the chat. This question is from Anoop. It's called, isn't FOXXP expression a requisite marker of Treg induced or otherwise? And I'm sorry for butchering all of these biological terms, by the way. Obviously, I'm not a subject matter expert.
So I think this is a bit more complex than I probably can get into, but when we think about what a T-regulatory cell is, we have to think about, all right, there's that sort of traditional CD25 positive, FOXP3 positive CD4 T cell that we call a Treg. But is there a regulatory regulatory sort of phenotype that goes beyond the fact that something's a CD4 cell? We know, in fact, that regulation is a product of both receptor expression as well as cytokine secretion. And we know, for instance, that some CD8 cells can fulfill this, and indeed, there's regulatory aspects of innate lymphoid cells for that matter. So I think FOXP3 is important for particular regulatory T cell types, but I don't have the data, but I believe at the time I was doing this, we found cases where FOXP3 was not necessarily expressed in all T cell types that might fulfill a regulatory function. But I'm willing to look into this more because we want to, of course, always have this as correct as possible. Go ahead.
Thanks so much, Alex. Next question is by Neil Kelleher. I'm also going to post that into the chat here. With modeling, have you been able to get an estimate of where the number of total human cell types will level off, 3,000, 5,000, et cetera, et cetera?
That's a very good question. I don't really want to speculate on that because, for instance, when we talk about epithelial cells, I mean, there's a general morphological pattern, morphological appearance to epithelial cells. There's types of surfaces that you find epithelial cells, but we have to decide at some point all the epithelial cells on a surface, are they all identical to each other, or if we can start identifying, sort of small differences in gene expression, are they really different cell types? And so I think eventually every cell type is its own class if you look deep enough, but at the same time, it's important to recognize major groupings, and it's just a matter of how granular do you want to get. I don't think there's a good answer to that question. It depends really how detailed you want to be. Go ahead.
Neil just put in another text to the Q&A chat, and he said, "Please speculate." Smiley face. So.
Well, oh gosh, I don't know. I haven't gone off in this direction in terms of my thinking. I did have some slides a few years ago I used to include about the number of cell types, but you know, when we consider all the variants of T cells, it seems every time you add a few markers, you might find something that really seems biologically significant. And so it's tricky. I mean, I think thousands for sure. I'm not going to say tens of thousands, but anyway, that's as much as I'm going to be pinned down here.
Thanks so much, Alex. All right, we have yet another question by Zarina Gallis this time. I also put it in the chat here. Is the amount of information available about each cell type in the cell ontology mainly data-driven, such as data availability, et cetera, or user-driven?
I would say the fact that we represent certain cell types is often user-driven, but the information available is often data-driven. But it's also driven by the amount of effort that you know, those of us who developed the ontology have had to sort of look into particular cells, meaning that we recognize that there are certainly cases where we can add information to the cell ontology about particular cell types that would be very useful to our users.
Thank you very much. All right, we have one last question by Ellen. Can you comment on the level of biomarker information linked to cell types and biomarker representation beyond immune cell markers assigned to immune cells? Or will this biomarker information be captured in a different place or way?
So again, our goal here is to define the necessary and sufficient criteria needed to uniquely identify one particular cell from all other cells in the body. In some cases, that may include biomarker information, you know, beyond representation of proteins that may be expressed. I mean, it already includes, for instance, participation or the ability to carry out certain biological processes. So we say that the regulatory T cells are capable of negative regulation of immune responses, and we use the gene ontology biological process term to reflect that. But you know, really general information about biomarkers and other aspects, I mean, full range of gene expression should be captured in a knowledge base such as HuBMAP. And typically, we hope that will be linked to the cell ontology as metadata, but the cell ontology can't capture all aspects in its definitions. So if it's needed, it'll be there. If not, it won't.
Thank you so much, Alex. This marks the end of the session. Thank you so much, Alex, for your fantastic talk, and also thank you for braving this Q&A method, which is really challenging, where you speak to 100 people and you don't see them. So yeah, this was really a fantastic hour that we spent with you.
I have a little present also for Alex, so you will get a little token of appreciation for your virtual talk to the HuBMAP demo day. And thank you so much. Again, it's our demo day, a trophy. Thank you.
Thank you very much. It's been a pleasure speaking to you.
Wonderful. All right, folks, we are now moving back to the home base, and then there will be further instructions there. So you can leave this webinar here and just return to the home base. You will see the link on the agenda. See you there in a minute. Bye, Alex. Thank you.
Bye-bye.